So now we're going to look at microscopy. When we're looking at microscopy, we need to also look at units of measurement. We do use the metric system. It's used and it's easy to use because it moves by powers of 10. So all you have to do is move the decimal for conversions when we go from one unit to the next. So if we go from 1 centimeter, it would go to 10 millimeters when we move through. Microorganisms, though, guys, are very small. We can't even use 1 centimeter or 1 millimeter when we're talking about these. We have to go even smaller. And so when we look at this, we may use micrometers. These are a millionth of a meter, or 10 to the negative 6. Or we may even use nanometers, which is a billionth of a meter, or 10 to the negative 9. So when we're measuring a lot of times these microorganisms, we have to use an ocular type of measurement instead of just using a normal like ruler. Now this brings us to microscopes. Since these things are so small, we've got to use something that's going to magnify them so that we can view them. So reviewing really quick, microscopes were developed by individuals like Hooke. He used the compound scope to study cells. Leeuwenhoek in the 17th century used one lens, which was a simple microscope, and he magnified those little animalcules. He thought they looked like little animals 300 times their normal size. Now there are lots of different types of microscopy, and they do get categorized by their energy source. There are light, photon, acoustic, electron, and scan probed. We're going to focus mostly on the light microscope because this is the one that you use most of the time in a microbiology lab. However, electron microscopes could also be utilized. So when we look at a light microscope, this is going to have some different variations, and the first one is what we call a bright field light microscope. This is where the specimen is light against a bright background, and we can actually use a wet or stained prep in order to view an organism. Now when we look at a wet mount, guys, this is where we take the organism, we place it on the slide with water, and we look at it that way. This allows it to potentially still be alive and moving. On the stained side, this allows us to see structures better, but the organism is dead. Now in lab, we use the compound light microscope with a bright field illumination, and so this is an important microscope that you need to learn the parts of. So you need to learn the parts and you need to learn their function. Some really important parts that are on these compound light microscopes is the fact that when you are using them to magnify something, you have to take the magnification from the ocular side, okay, so the part from where you put your eyes, which is always about 10 times, they're magnifying 10 times, with the objective. Now the objectives are found in the middle of the scope, and they're the ones that move that we can turn up or down based on what we want to see. And so some objectives are going to be like a 4 objective, so we would take 4 times the 10, which are the visual, and that would be 40 times. Let's just say that it starts as a 40, well then 40 times 10 is 400. If it's the oil, it's 100 times 10, which is 1,000 times bigger than normal. And so those are the 2 areas when we talk about compound that are going to magnify what you're seeing. The other part is because this is a bright field, it needs to have a light source that's present, and that's also shown here in the picture. But guys, you do need to make sure that you review the parts of the microscope and you understand what they do, because even though you may not be utilizing one right now, there might be a chance that you come in contact with a microscope like this. Another type of light microscope is called the dark field light microscope. This is where the specimen is light against a dark background. We use wet mounts for this, and the whole point of this type of dark field microscopy is to detect motility or movement. So if you go in and you click on the video that's listed here, you can see the spirochete responsible for syphilis, and you can see how it moves on that microscope. Another type of light microscope is called the phase contrast. This is where they use filters, and these filters are going to alter how the light is refracted and diffracted, and we can use wet preps or wet mounts here, and this allows for internal structures to be viewed. And so I did a comparison here for you. It's the same type of organism, and you can see it on the bright field microscope, a dark field, and then the phase contrast. And even though it's the same organism, you can see different structures based on what kind of light microscope is being utilized. Another kind of light microscope is called a fluorescence light microscope, and this uses ultraviolet light source like a black light, and they're going to view fluorescence against a dark background. Now, you can use a special, what we call fluorochrome stain, and this gives some fluorescence so that it can be viewed. Some are naturally fluorescent when we look at certain microorganisms, but some stains have to actually employ something a little more complex, which are called tagged antibodies. These tagged antibodies are specifically going to target an antigen on that organism, and this also helps us ID that organism. And it shows you a picture here where they take an antibody, they tag it with the fluorescence label. When the antibody attaches to the appropriate organism, we see the fluorescence that is present. Okay, and so it's showing you again how it kind of lights up in this type of microscope. For the electron microscopes, we see that electron microscopes are very helpful because they give you very high resolution. We have two kind of categories of electron microscopes, and one is the transmission, or the TEM. You've got to stain the internal structures so that they can be viewed through this type of electron microscope, so it kills the organism. But the magnification can be 10,000 to 100,000 times its normal size. The scanning, or SEM, electron microscope uses 3D surface features, so it can magnify not as great, but it can show more surface features. This also allows the organism to pretty much stay alive during the process as well. This magnifies 1,000 to 10,000 times. Now, how do we decide what kind of microscope needs to be used? Well, first of all, you've got to understand what you are studying. Okay, what are you going to be taking a look at? Does the organism need to be living? If it needs to be living, this might limit which type of microscopes you could use. Do we need to stain it so that we can see special structures? How much magnification and or resolution do you need? Okay, so what happens a lot of times when we look at this, guys, is as magnification increases, we make it bigger and bigger. More resolution, though, is required in order for us to be able to see two distinct points. Okay, so an example here, here's the boot, and if you want to look very closely at a particular structure, we can zoom in and that magnifies it, but it doesn't give us a good resolution. Do you see how it looks all fuzzy? We need to make sure that we can magnify, but also be able to focus the image, get a good resolution. And so this is, again, what's going to be helpful when we're deciding what kind of microscope to utilize. All right, so what type of microscope would you use to study the following? If we look at macromolecules are like proteins and lipids, you would have to use an electron microscope. If you're going to want to look at viruses are mostly electron microscopes, but they are on the very tip of a light microscope. Bacteria, protozoas, some types of your fungus, those are going to be more of the light microscope. Some fungi can actually be seen with the naked eye. Same thing with the helminths or the animals. They can be looked at with the naked eye or through a light microscope. Okay, so these are what we're using. So the light microscope is kind of the one that's the most important here in microbiology, because you can see most of the structures of what we're wanting to look at in that category. Sometimes when we want to actually be able to look at a specimen, we're going to have to stain it. This is going to mean that we can see and distinguish parts of it a lot easier with the staining. In some cases, staining might not be needed, and so this would be what we would call more of a wet prep or wet mount on a slide. So how to create a wet prep or a wet mount is you would take your slide and you would drop some water with your specimen in it onto the slide. You're then going to place a cover slip. The cover slip will spread the water out, and then you will then view it underneath the microscope. Some advantages to this is it is quick and easy, and it does allow you to detect motility, movement. The problem with this, though, is there's no permanent record. Once you take that off, all those microorganisms that were on that wet mount are gone. It's also very difficult to see specific cellular structures or details when we're using a wet mount because there's no stain involved. So the reason we do staining is to give color to the specimen against that bright background so that we can see things a little bit better. All right, and so again, here's a link to a video if you go into the PowerPoint and you can look at some examples of motility. Now, can you use the oil ejective viewing a wet mount? This is a pretty good question, and the answer is no. This is not a common practice. If oil is used, the slide has to be sealed first because it's going to cause some movement. And remember, oil and water do not mix. Okay, and so this could actually cause it to be more blurry and you not be able to see the details. So when we do need to stain a slide in order to see things a little bit better, we need to prepare our slide with what we call a smear. So how do we prepare a smear? Well, you're going to mix a small amount of the specimen that you want to view in a few drops of sterile water. Okay, so we want to use this sterile water, and the sterile water means that there's nothing else in it. So the organism we're wanting to be able to view is the one that we're adding. We also see that you could drop some broth that has the organism in it as well. If you know that it's not contaminated, you could use that. Now, one thing that's super important when you're creating a smear is you need to let it air dry. Okay, so it needs to air dry, and then you're going to heat fix it. We heat fix it by adding heat. This helps kill the organism, and it also helps the specimen stick to the slide, okay, because we're going to be staining it with lots of liquids and we don't want it to wash off. Okay, otherwise then what was the point? Because then you can't see it anyways. So we need it to stick to the slide. You then have to let the slide cool completely. All right, and this is hard sometimes because people normally get impatient when it's the whole let it dry, let it air dry. They get impatient and they want to just heat fix it right away, or let the slide cool, because when we're talking about a glass slide, it may look like it's cool, but it might not be. Okay, so it's really important to let it cool completely. Once it is cooled completely, then the smear is ready for the staining process. Now, some sources of error that can occur when you are preparing a smear. Your smear might be too thick. You put too much of the specimen on one little area. Also, your heat fix happened before you let it air dry. Again, we talked about how we sometimes get impatient. Or you heat fixed for too long. If you heat fix for too long, it distorts what the cells look like. Now, some overview of some stains. We see that we can use some basic dyes like positive ions and also some acidic dyes like negative ions in order to stain different types of organisms or specimens. Bacterial cells, however, have a slightly negative charge when they're in about that pH of 7, and since they have that slight negative charge, which type of dye would be attracted to the bacterial cells? Now remember, back when we talked in the chemistry chapter, what attracts? Opposites attract. So if the bacterial cell wall is slightly negative, we need to use a positive dye, or what we call a basic dye. Acidic dyes won't work as well because they'll repel, okay, because a positive and a negative and negative will repel each other. So some common basic dyes that are utilized in the lab are methylene blue, crystal violet, saffronin, and what we call malachite green. So these are just some common basic stains that are utilized. When we're staining, we might utilize what we call a simple stain. The purpose of a simple stain is to determine the shape and arrangement of cells. A very common simple stain that's used is methylene blue. Okay, when we use methylene blue, it stains the cells a kind of bluish color, and it allows you to be able to see their shape and arrangement. Like in this case, it shows you how staph is different than E. coli. You can see that there are structural differences. The shapes are different. Whereas staph's going to have more of the cockeye, the round shape, as well as when we're looking at this, they're in clusters, whereas the E. coli is more in a rod shape that we call bacilli or bacillus. We may need to take staining a step further, and these are known as differential stains. Differential stains are going to allow you to distinguish between different types of bacteria by what we call a Gram reaction. This means we're going to be making it where we can put them into groups. Instead of just looking at their basic structure, their basic arrangement, we can actually differentiate them. Now, the Gram stain is one that's really important. It's one of the most commonly used to ID bacteria, and this either puts that bacteria into one of two categories. They're either Gram positive or they are Gram negative. So let's talk about the process first, and then we'll look at what those kind of results look like. So when we do Gram staining, we're not just putting one stain like we saw with the simple stain on it. We're going to do a series of steps, and it's important to complete the steps in the appropriate order. So we'll first start with the primary stain, which is the crystal violet. So you would take your smear and you would put the crystal violet on there. You let it sit for about approximately 60 seconds, okay, for about a minute. Then you are going to rinse the stain off. you'll notice that there's going to be purple still within your smear. You're then going to use what we call mordant. The mordant in this case is iodine. You'll place the iodine on it, flooding the slide, and you'll let it sit for a minute. Once you've let it sit, you're going to then rinse it. Guys, the mordant allows for the gram-positive bacteria to take in that primary stain a little bit more and hold on to it. You're then going to use a decolorizer, and guys, this does exactly that. It decolorizes, it takes the color away. This is the one that has the most critical timing that you have to pay attention to. This is 95% ethanol. You're going to place it on for just a few seconds. It's not going to sit there for 60 seconds. If you do that, it actually will rinse all the color away, and the staining process is just void now. Okay, so you only leave it on for a few seconds, and then you need to rinse it off quickly. The secondary stain then is called the counter stain. This stain in this particular case for gram staining is the safranin. Okay, you'll put the safranin on and flooding the slide, let it sit for about a minute, then you'll rinse it. Now the staining process is done and you want to blot it dry. We don't want to wipe it dry. If you wipe it, you're going to end up wiping off your smear. We want to blot it dry, and this will then allow us to get ready to see it underneath a microscope. Now, the principle behind gram staining is that when we look at the staining process, crystal violet is going to stay if it has a thicker cell wall. This means it's a gram-positive bacteria and it will have a purple color. They have a thicker peptidoglycan layer in their cell wall, so the mordant, in this case the iodine, drives the crystal violet into that cell wall, so it can't easily be washed out by the decolorizer. Now, if you leave the decolorizer on too long, it can be washed out. So this is why it's really important, that timing issue. Okay, because it could lose that color purple if you decolorize for too long. If not though, if you just do the decolorizer like you're supposed to, it will remain purple. On the other hand, gram-negative bacteria will appear pink. They have a thinner layer of peptidoglycan, so the mordant does not push that crystal violet in. The crystal violet actually gets washed away during the decolorization, and so we lose that purple out of the cell wall, and then the counter stain, the safranin, is what is held on by that cell wall, so they appear pink. So gram-positive will appear purple, gram-negative will appear a pinkish red, or pink or pinkish red. So let's take a look. If we are to do this staining process again, what's happening at each step? So after the primary stain, we've added the crystal violet. Okay, what color are the gram-positive and the gram-negative? Well, they're both purple, because that's the stain that's been put on there. Nothing else has happened, so they're both going to appear purple. So this does not allow us to differentiate anything. We've got to continue with the steps. After the mordant and the decolorizer, this is the key. It's the iodine and the alcohol, the decolorizer. After that step, we see the gram-positive are still purple, but the gram-negative now are colorless. We've washed the color away. So now the colorless bacteria is ready for the counter stain. So after the counter stain's been added, what color are the gram-positive? Well, they're still purple. Okay, because they still have that crystal violet in their cell wall. The gram-negative, however, are now a pinkish red. Okay, and so we see this is why we have this specific structure with gram staining. So if we take a look here, we can see that we have some gram-negative bacillus. You can tell it's gram-negative because it is a pinkish color. On the other hand, in this same picture, you're going to notice that there's these purple long structures, so there's also gram-positive bacillus. Guys, this tells you that there are two different species of bacteria located here because they gram stained differently. On the other hand, in the second picture, you can see that there is a gram-positive, but this gram-positive has got the cockeye shape. It's round or circular, whereas the gram-negative here is still a bacillus. Okay, so we can see that. Now, what about this last one? This last one's hard because you see some areas that are purple, you see some areas that are pink, and you see some areas that are that kind of in between the two. This could be a gram variable bacillus. Okay, it could be. There are some of them out there. Gram staining doesn't work the same all the time, and some bacteria have some exceptions to how gram stains work. But these could also be old cells. If we take old cells, then they do not stain as well. Okay, so this is why in the lab it's really important to have new cultures that you're dealing with. Also, they could have over decolorized the gram-positive or under decolorized the gram-negative. They could have messed up that critical step with the decolorizer. We would need to do a new smear and a restaining to see which one of these is technically true. All right, and so this is where, again, some of those sources of error could come in. Another type of differential stain that can be used is called the acid-fast stain, or also known as the Z-Nielsen or the Kenyon-Cold method chemical mordant stain. These are used to ID what we call mycobacterium. One of the main examples of mycobacterium is tuberculosis or TB. We also can see that these could be used for what we call Nocardia species as well. So these are what we would use. We can't use gram staining for them, so we would use an acid-fast base staining, so an AFB stain. Now again, this particular staining process has steps to it, and the steps need to be followed, but these steps are a little different than what we saw for gram staining. So we see first there's an application of the carbofusion stain. That's the primary stain, okay? And it shows up that it's all kind of this pinkish color. We then see the mordant in this case is not iodine, it's actually heat. So you would heat up the stain. Then you would have the application of the acid alcohol. In this case, that's the decolorizer. Once the decolorizer has done its job, you're going to then have the application of the methyl blue or the counter stain. So we do see that it's going to have the acid-fast positive bacteria is going to be the pink color. The non-acid-fast bacteria would be the bluish color. Now the principles here is that microbes with a high lipid or what we call the mycolic acid content is going to actually retain that carbofusion stain even after the acid alcohol wash. And again, this allows you to differentiate and be able to figure out which kind of bacteria you're looking at. There's also some other special stains that are out there, and these special stains, the whole purpose is to stain particular bacterial structures that we want to see. One example are called negative stains. These are also known as Indian ink or what we call nigrosin stains. They're going to detect capsules located around the outer portion of a cell wall. This is a simple staining technique, but you'll notice when you see a capsule present, it causes there to be this kind of decolorized area around it. Capsules are helpful in helping with virulence and things being a little more pathogenic sometimes. We also see endospores could be potentially stained. This is going to be through what they'll call the Schaefer-Fulton method. Endospores protect bacteria from adverse environmental conditions. It causes them to kind of go into a dormant stage. Okay, so they go into that dormant stage and they're just waiting for better conditions so that they can make somebody potentially sick again. And this is a differential staining technique as well. And you can see it, the vegetative cells are pink, but the endospores are stained in this bright blue color. We also see that we could use the flagellar stain. This is going to stain the flagella. This allows us to see what kind of motility the type of bacteria might use. Now, all of these structures are going to contribute to the virulence or the pathogenic factors of the bacteria. When they have some of these structures listed here, it causes them to be able to overcome some of maybe our immune system, some of those other things that help keep it out of an organism to where it becomes a little more pathogenic.